So I went here for basically sophomore year through senior year. Moved back for 2 years of college and now I'm here.
Vulcan Lake has a college. That's where I learned to water ski. I love Folsom Lake, except that in later years, you can't go. Because there was no Folsom Lake.
There wasn't really good.
The Folsom Lake, the Merced River was dry. There's just nothing.
It got so low at one point they found, on Folsom Lake, they found a lot of old things and a couple of dead bodies.
Oh, good. Yeah, you should empty a lake periodically and sweep it out and then fill it back up with fresh water.
It's good now, except there's a huge controversy because last year they dumped out more than half the water in Folsom Lake. And
Yeah, into San Francisco Bay.
Yeah.
Oh my gosh.
So everyone was pissed because rain is, it's rare. It's
Yeah,
rare
our last
in
visit
California.
to Mariposa, you couldn't take showers.
Mhm.
It was like, what is this, Phoenix?
And we had fines for every time we went over like a certain water usage.
Uh-huh.
Yeah. I can't see why they, find a tunnel system to transfer water from different places. That'd be a good thing to do.
What
Yeah.
they do. We got good ones like rivers.
Yeah, but not to California. And there was one time when rivers got so low you could walk straight to the other side. It was not a good 10 years for California. What is that? Do you hear that?
No,
Somebody
that's-
upstairs. I don't know if you noticed, but every once in a while the projector bounces around. So it's on the screen will fidget. Oh, it's somebody writing on a whiteboard.
Yeah, really forcefully. Really forcefully. Okay.
I found out how to get rid of the glare, so this will represent the first video up there that you can actually see what's on the board. So
Assuming, of course, you can actually see it until it gets dark. And I don't know why that always takes so long to warm up enough to blink to a bright screen. All right. So let's talk about Alzheimer's for a little bit. Alzheimer's is a neurodegenerative disorder that initiates symptoms similar to dementia, leading to memory loss, inability to handle daily tasks, and eventually shutdown and death.
that's what I.
Yeah. So, what's that like? What causes it? In the brain of a normal person, you have small beta amyloid proteins, which is breaking the own rule of the book here, made up of 42 amino acids. But normally, they form alpha helixes. But when you have Alzheimer's, instead what happens is instead of being these bent alpha helixes, they reform to make beta sheets. Those beta sheets do not have the normal function. And in fact, those beta sheets are not soluble in solution. And so what you get are the plaques, these neurofibrillary plaques, beta amyloid plaques that form inside the cell bodies of neurons. And the problem with this is that once this process, see there's the blink, once this process starts, it is self-perpetuating. Because once you get these misfolded beta sheets, these misfolded proteins, they encourage the misfolding of other proteins to form those stacks that you were asking about, right? And so once you have these types of stacks, they will associate with the normally folded proteins and those proteins will change shape in order to form a beta sheet and add on to the plaque.
So it's almost like it's magnetic.
Yeah. It's not, yeah, I mean, as a metaphor, yeah,
Right.
it's sort of like it's magnetic. The proteins will, the actual word is chaperone. Having these proteins will chaperone the misshape or the misformation of other proteins like them to just lead to a exacerbating the problem where it just kind of keeps going and keeps going and keeps going. And as these plaques build up in the cells, they limit and or kill off the ability of, or destroy the cell's ability to propagate signals and to function normally. Now, how many of you guys have heard of mad cow disease? So have you heard of a prion disease?
Yes.
That's what mad cow disease is. It's a prion. Now, when we talk about infectious diseases, we usually talk about infectious diseases coming in the form of either viral or microbiotic, i.e. bacterial, protistan, fungal. It's just some of the living organism versus viral, right? Well, there is a third class of contagion, if you will, that is not viral and is not fungal or cellular in any way, and that's prion disease. It's proteins, small. They have to be small because they have to be able to survive going through your digestive tract and not being destroyed. If they're really, really large, somewhere in there they're going to get cut by your digestive system, and therefore they will be non-functional and will be harmless. But small, certain, very specific, small proteins can survive the digestive system. Mad cow disease is one of those. If a cow is fed the protein, specifically brain matter of another cow, and that cow had any of those misfolded proteins, then the new cow will develop mad cow disease as well, because those proteins will be absorbed by the body. Eventually they will get into some of the neurons, and then it will cause the misfolding of the normal proteins that were there anyway in that cow, leading to more of these small protein fragments, and therefore if that cow then is fed to other cows, the disease will perpetuate. Is there such a thing as a prion disease in humans?
Oh yeah.
Yes.
Crutchfield-Jacobs, spongiform encephalitis.
Prion diseases are rare, but they do exist. And there are even known examples of prion diseases that affect the brain, effectively mad cow in people. There was a group near the tip of South America, if I recall correctly. One of their rituals that they did when someone died was they would bury the body, but they would cut out, I believe, the heart and the brain, and their relatives would eat it as a form of carrying their relatives with them. This was not healthy, and led to the development of neurological prion-related disorders within those populations. And understandably, when you are sharing the brain meats with multiple family members, it was somewhat disastrous. Point is, it's all due to the folding of those proteins. Beta sheets versus alpha helixes. That's what, I don't want to use the word causes. That is the thing that we use as a measurement for Alzheimer's. But Alzheimer's is not a prion disease. You don't get it by eating the brain of your great uncle that had Alzheimer's. But we do and are searching for links, genetic or otherwise, that increases the risk for Alzheimer's. What can cause this to start? And therefore, once it's started, it starts running amok. What can we do to eliminate those beta plaques? To potentially cure Alzheimer's disease, or at least stop it from getting any worse? Those are the areas of research for Alzheimer's. So when I don't want to say that these plaques are what cause Alzheimer's. These plaques are the thing that cause the effects of Alzheimer's. We don't really know what causes them in the first place. At least not to my knowledge. I'm not an expert on Alzheimer's research.
But the plaques are how we actually are
Plaques
the
are how we-
diagnostic too.
Yes, the plaques are the diagnostic measurement. Beyond simply the reason that someone would go in to be asked, and that's dementia-related disorders. Dementia-related symptoms. So that's secondary structure. Alpha helix, beta sheets, and yes, They can convert back and forth between the two under the right conditions. But in either case, what is it that determines whether or not it's going to be an alpha sheet or a alpha helix or beta sheet? It's the primary structure. The primary structure determines the secondary structure. Now, we can force it to go one way or another. That's the whole point of the Alzheimer's beta flags. In tertiary structure, we are going to take that secondary structure and then now start associating those secondary structures with themselves. So the important differentiation between secondary structure and tertiary structure is, here is where we start involving the R groups. Here's where we start involving the special chemical nature of those side groups into how the protein takes shape. So, here we have some beta sheets that are associated with each other. They might be associated and they might be hanging out here because of hydrophilic interactions with some of those side groups. Here we have an alpha helix that's going to bond with another hydrophobic interaction down here with some of this side chain hanging off to the bit. Those guys are hydrophobic and are going to avoid water and hang out together. Here we have some disulfide bonds between some cysteines, some of which in this alpha helix, some of which in these beta sheets. Those two beta sheets are associated with each other because of hydrogen bonds stacking on top of each other. And then we could also have some hydrogen bonding here between these side groups and some salt bridges over here between some positive and negatively charged amino acids in those two alpha helixes. But the point is, what determines the tertiary structure of this protein? The nature of the side groups, right? But those side groups force this particular shape of this protein because of the order where you find them within the secondary structure. So in other words, the tertiary structure is dependent on the secondary structure. And we have already established that the secondary structure is dependent on the primary structure. So ultimately, everything comes back down to the order of the amino acids. The order of the amino acids is going to determine where we get our alpha helixes and beta sheets. That is going to determine how they're going to bend back on each other because of the association of the R groups in those alpha helixes and beta sheets. By way of example, if I were to change that amino acid from a positive amino acid into a neutral one, or even worse, another negative one, what would that do to the shape of this protein? If it's neutral, we wouldn't have any reason for these two guys to hang out, right? If it was negative, we would have reason for those two guys to actively want to be apart from each other, which would give us a protein with a different shape. Tertiary structure is the association of the R groups. Secondary structure is the shape we take to stabilize and is stabilized by the backbone. Hydrogen bonding of the backbone. And all of that is dependent on our primary structure, the order of the amino acids that we used in the first place.
So the background get broken in the tertiary?
Can the background get broken? Mm-hmm. Yes. Mm-hmm. Okay. Now it doesn't, allow me to rephrase. The backbone can be broken. Yes. Insulin, by way of example, starts as one peptide that then bends back on itself, cross-links, and another enzyme cleaves it so that we have the two chains. It starts as one long chain, but another enzyme cuts it. So it's not that it bends back enough to where it breaks or snaps or something, but yes, it can be broken later. And in fact, a lot of enzymes come with off tabs, effectively, off or on tabs. A lot of enzymes are made in a shape that doesn't work. And then it's kind of like having a race car that's just sitting at the starting line. The engine's not actively revving, right? It's waiting. It's not going anywhere. And then something gives it the signal to step on the gas, and then off it goes. Proteins frequently have extra bits attached to them, and as long as that bit is there, they don't work. But if something else comes along and snips it off, then we have an active form that goes and does something. Or we could be waiting for something to be added to it, and when you add that thing, it causes it to change its shape, and now it's suddenly active. And if we take it off again, it'll turn it off again. There's a lot of things like that. So yes, there's what's called post-translational modifications. That may not mean anything because we haven't described what translation is yet, but we'll get to it. Sort of, maybe.
So like if your ribosomes go nuts?
Yes, translation is when your ribosomes make the protein. That's translation. Hydrophobic interactions can hold things together when we get nonpolar amino acid side groups, which would be found on the amino acids that we have labeled as nonpolar, right? But remember, all amino acids are soluble, all of the 20 that we care about are soluble in water. It's the side groups. So get a bunch of those together, and they're going to want to avoid water. You get hydrophilic interactions where you have those polar or positive or negatively charged groups that really want to hang out with water. You can have salt bridges where we get positive negative associations, hydrogen bonds between the side groups. Important here, hydrogen bonds between the side groups and not the backbone. Hydrogen bonds in the backbone gives you what? Secondary structure. And you can get disulfide bonds and other covalent linkages. All of these things help to give you the tertiary structure. So far, everything that we've been describing has been the shape of one protein, one chain of amino acids. It could be a really long one that forms a bunch of different beta sheets and alpha helixes. But if we start right there, we go wrap around here, a beta sheet, another beta sheet, then an alpha helix, a bunch of bits that don't take any particular shape, then an alpha helix, then a beta sheet, then another short helix, then a beta sheet, then another helix, then another helix, then a beta sheet, then another helix, and then we're done. All of that is still just one chain of amino acids. Tertiary structure dependent on secondary structure, dependent on primary structure. But the thing about proteins is we still have one more step, and that's quaternary structure. We haven't ever had a 4-0 before.
Should we be concerned? Some of it's cut out.
Yeah.
Well, you weren't responsible for that content, but now you are. I'm just giving you a hard time.
All the time.
Here's the important bit. What makes quaternary structure a quaternary structure? The short version, the TLDR version is you have to have 2 polypeptides. 2+. Does every protein have a quaternary structure? No. Not every protein comes as part of a multi-protein complex. Some proteins do a perfectly good enough job all by themselves. The example that's given here is hemoglobin. We have 2 alpha proteins and 2 beta proteins. But the point is that the job of hemoglobin is to hold on to this heme group. It's a ring structure with an iron atom in the center of it. A planar ring structure with an, well, mostly planar, with an iron atom in the center of it. But that is held together in the gaps. Not so much the gaps, but in a junction point, if you will, between an alpha and a beta. We have heme here, heme here, heme here, heme there. An individual hemoglobin protein, like heme A and heme B, by themselves, they can't carry oxygen. But when they form a tetramer like this, they can hold on to multiple heme rings, and therefore now they can carry oxygen. Four polypeptides coming together in a very specific shape. So, what is it that determines these 2 things linking with those 2 things? What's holding this red group to that orange group?
Tertiary structure.
the same things that held the tertiary structure together. It's going to be hydrophobic interactions, it's going to be salt bridges, it's going to be covalent linkages. Something is holding those two guys together. But once again, if we change the primary structure, then we could change with the parts that actually cause them to stick together, which would mean that maybe they wouldn't stick together anymore, which would mean that they couldn't do their job. Stabilized by the same interactions found in tertiary structures. Not every quaternary structure is going to have 4. Some of them are just 2. Some of them are 12.
That's not confusing.
No, it's not. Which is why I told you at the beginning of it, the thing that makes quaternary structure a thing is when you have 2 plus. Primary, secondary, and tertiary is all talking about one protein. Every single one of these guys has a primary, secondary, and tertiary structure. The quaternary structure of hemoglobin requires all 4 subunits, or it's not complete. So, once again, primary, the order of the amino acids. Secondary, how they fold and hold on to each other on their backbones. Tertiary, how that folds and hangs on to each other based on the R groups. And then quaternary, if it requires more than one protein, then we show how those guys hold on to each other too. There is no way that you could actually quantify this. I can't ask you what holds together the tertiary structure of hemoglobin and expecting you to be able to say, well, it's the hydrophobic interactions of amino acids 237 or something like that. You don't need to know that. What you need to appreciate is that all 1 through 3 is the same protein, 4 requires multiple proteins, but all of them are dependent upon the step that comes before them. Ultimately, all of them dependent upon the order of the amino acids that was used in the first place. In the same way that how you spell a word depends on the order of the letters that you use in the first place, right?
The components of a tertiary structure are all elements that are, I'm sorry, not tertiary, quaternary. The components of a quaternary structure are all things that are in a tertiary structure?
They're the same thing. The things that stabilize a quaternary structure are the same things that stabilize a tertiary structure. So the reason these 2 alpha helixes hang out together is because of this salt bridge and that cross-link. Well, if you just make those 2 alpha helixes part of 2 different proteins, now we're talking about a quaternary structure association instead of a tertiary structure association.
But I mean, could you have a mix and match, like one of the proteins is in secondary structure? And...
No,
okay.
Because it's not going to stop. Protein isn't going to stop at secondary structure.
Okay.
It's going to fold all the way into whatever shape it takes by itself. Now, when it bonds with another protein, that might change that shape a bit. But it's not going to be like my tertiary structure is associating with your primary structure. It's just how they interact once they are complete proteins. That determines quaternary. But it's the same molecular mechanisms, the same salt bridges, the same covalent linkages, the same hydrophobic, hydrophilic interactions. It's the same thing that held them together in tertiary. That's the same type of forces that are holding them together in quaternary. So linked to health, this is a somewhat ubiquitous example that is discussed, and that is once again where we started from. I mentioned it passingly, sickle cell anemia. This is just a preview of the normal beta hemoglobin side chain. Certainly not the entire thing. But in normal beta hemoglobin, you get a glutamate right here. Proline, glutamate, glutamate, lysine. In sickle cell anemia, it goes proline, valine, glutamate, lysine. We have changed one amino acid. Now we haven't talked about this yet because this is a chemistry class, but next chapter we're going to talk about DNA. Do you know how many nucleotides it took? How many mutations did it take to go from normal red blood cells to sickle cell anemia red blood cells? One amino acid, so it can't be that many, right? But the answer is 1. Out of 1,500 nucleotides that it takes, 1,500 A's, C's, and G's, to make the normal beta hemoglobin side chain, one of them changes, and you get sickle cell anemia. Now, I ran a molecular genetics lab for 10 years. I could have gone in and manually altered probably 700 to 900 of those 1,500 A's, T's, C's, and G's, and it would have produced exactly the same protein. And I don't mean it would work the same. I mean it would be the same. The reasons for that we will better understand next chapter. Sort of. This is chemistry, not biology. But the point is, what's it take to make a potentially lethal disease? Sometimes as little as 1 amino acid out of 500. 1 amino acid out of 3,000. 1 amino acid out of 10,000. Sometimes that's all it takes. Why? Why would that make a difference? Glutamate is a polar acidic amino acid. Valine is a non-polar amino acid. What's a polar acidic amino acid going to do?
Form a salt.
It could form a salt bridge. It's a polar acidic amino acid. 1, associates well with water. 2, it is charged at physiological pH, and therefore it can form positive, negative associations, or positive, positive, negative, negative repulsion, right? What about a non-polar amino acid? Is that going to get along well with water?
No.
No. Is it going to associate with a positive or negative charge? No. You could change a lot of things around and not change much. An alanine to a glycine is not going to make that much of a difference. It's a non-polar short-chain amino acid to a non-polar short-chain amino acid. It's probably going to change nothing. But you go from a glutamate to a valine, and that's a really big change. It still could have no difference. But as it turns out, it does. Normal hemoglobin forms this nice tetramer, 2 alphas, 2 betas. If you have sickle cell beta, if you have sickle cell anemia and you have beta chains, then what happens is those nice tetramers will start forming longer chains. Tetramers will bind to tetramers. And you'll get these long chains. And those chains will eventually form even longer insoluble fibers. It decreases the ability of those red blood cells to carry oxygen. It changes the shape of the red blood cells in general. It can clog capillaries, which means that their tissues won't be able to get the oxygen that they need because the blood cells won't even be able to get into those tissues. Where do we find sickle cell anemia? At the beginning of the chapter, the example that was given was an African baby adopted by American parents.
It has sub-Saharan, doesn't it?
Mm-hmm.
Okay.
Sub-Saharan African
Population.
origin. Here's the thing though. Sickle cell anemia is a disease that is endemic to, yes, sub-Saharan Africa. Guess what else is endemic to sub-Saharan Africa? Malaria.
And AIDS.
Well, a lot of other things are endemic to sub-Saharan Africa. Sickle cell anemia, if you're a heterozygote for malaria, your cells don't sickle unless they're under stress. Like when they're infected with a malarial parasite. A heterozygote. So if your parents were both heterozygotes, if they carried one normal beta hemoglobin gene and one sickle beta hemoglobin gene, that means some of the beta hemoglobin you make will be normal and some of it will be sickle.
Okay, but you said heterozygote
For
malaria.
No, excuse me, heterozygote for sickle cell anemia.
Thank you.
If you are heterozygote for sickle cell anemia, that makes you resistant to malaria.
Oh, yeah.
Africans were living in sub-Saharan Africa under the threat of malaria for literally thousands of years before we showed up, before medicine advanced to the point where we could treat malaria with drugs that are not always very cheap. Malaria is life-threatening. You can survive it, but it can also kill you, especially if you're very young or very old. So if you live in an area where there's this disease that kills people somewhat indiscriminately because Mosquitoes don't prefer 20-somethings, they don't prefer elderly, they don't necessarily prefer babies, other than the fact that babies can't get rid of them. So if you're born without any of these genes, then you are susceptible to malaria. If you're born with 2 copies of it, then you're probably going to die absent medical attention from sickle cell anemia and the problems that it brings with it. But if you're born with 1 normal copy and 1 sickle copy, then malaria is not so much a threat anymore. Now, there are still a lot of other things that could kill you, but now there's 1 less. So who's left to make most of the babies?
The people
The
with
people
sickle
who
cell
are
anemia.
heterozygotes, that are carriers of sickle cell anemia. Problem is that then that makes them at greater risk for having children that actually have both copies.
So, is there speculation that sickle cell anemia came about as a evolutionary
Yeah.
response to malaria?
Mm-hmm. It became prevalent in those populations as an evolutionary response to malaria. But for a long time, we also used sickle cell anemia as, it was called the black man's disease. Because what we were looking for was just more and more reasons why we should justify poor sociological treatment of a certain class of society.
More reasons to be an asshole.
Anyway, that's sickle cell anemia. How it works, why it works, and also why it was common, where it is common. The reason why I throw that extra little bit in there is because I like, as a biologist, to also impress upon you how not everything is as black and white as, is this a good mutation or a bad mutation? Sickle cell anemia is most frequently discussed as a disease, as a disorder, as a bad thing. And in modern society, sure it is. But previous to medical advancements, it gave heterozygotes an advantage. It also meant that 1 out of 4 of their kids died from sickle cell anemia. And 1 out of 4 of their kids might have died from malaria.
Sickle cell's not sex-linked?
Was that?
Sickle-
Sex-linked? No.
Because that would give you the malarial benefit and, you know, by...
Well, the problem is if it was sex-linked, then males would only express 1 version. So they wouldn't be able to be heterozygotes. And so males would either die of sickle cell anemia or die of malaria, unless they happen to live long enough without getting bit by a malarial-infected mosquito. So sex-linked wouldn't have made it better, it would have made it worse. Females would still have had both, and males would have only had 1. You only have 1 X chromosome, because the thing that makes us male is the Y chromosome that comes with it. So whatever's on that one, that's the only one we have. We can discuss that further after class. So, structural level of proteins: primary, secondary, tertiary, quaternary. Just for point of emphasis, even though I have already said it, quaternary is only when you have 2 or more protein subunits. Not every protein has quaternary structure. Some proteins stop at tertiary because they do their job all on their own. The quaternary structure of those misfolded amyloid plaques could have thousands upon thousands of subunits all stacked on top of each other, right?
Now, the subunit is a protein, not a subunit of the protein.
Correct. A subunit is a single peptide that goes to tertiary and then combines with other tertiary proteins to form a quaternary.
Okay. It's just how they grammatically worded it. That
Yeah.
Little ambiguous.
All right, so let's talk about denaturation. What is that? How often do you guys denature proteins?
Every Friday night.
Every Friday night? Depending. Sure, I would do it. I have a pet peeve about that, and if we have time, I'll tell you about it. This is your brain on alcohol. Ask me about it. Denaturation. If you cook, you denature proteins. That's what cooking does, is it denatures proteins. You're also denaturing proteins in your stomach every day after you eat. You're denaturing proteins in your intestines every day after you eat. You're denaturing proteins in every single one of your cells every day as a process of your normal biochemistry. What is the act of denaturing a protein? It's when we disrupt the bonds that give it its shape. Because something that has not explicitly been stated yet, but is going to come up, the function of a protein is dependent on its shape. Everybody knows what a hammer looks like, right? What makes a hammer a functional hammer? Well, we could say its shape. Let's take that hammer and throw it in a big furnace and melt it down. And when we're done, what we will get is this kind of weird pooled thing of metal, relatively thin. Will that still work as a hammer? You could all, well, what about the handle? The handle's gone. Okay, fine, I tape a bit of wood to it. It's the same thing now, right? It's a metal bit with a wooden bit. Is that going to be a very functional hammer?
It can.
If I try really hard, I could still maybe hammer a little bit of something with it, or like no, it's not.
You just step on it.
Yeah.
So it's
Hit
a
it really hard
denatured,
with your hand.
a denatured hammer.
Pick up a rock. Yes, it's a denatured hammer. Point being, it no longer functions as a hammer. The job of an enzyme, of any protein, its job is dependent upon its shape. If you change the shape, you change the function. Usually, the change is destroy the function. Sometimes it's give it a different function, but that's very, 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 very rare. Usually it's destroy. We can do this with heat. Why would heating it change it? Tells you the answer there, but
It breaks apart the hydrogen bonds and disrupts hydrophobic interactions.
right. Hydrogen bonds are what hold together what portion of the overall shape of a protein? There's 4 options.
The linkages.
Where? Linkages where?
In the sheets and also in the
Okay, in secondary
fields.
structure?
Thank you.
Is it involved anywhere in tertiary structure? Because it's hydrogen bonds between the backbone in secondary, but it's hydrogen bonds between the R groups in the tertiary, right? And could it be involved in quaternary? Same things that affect tertiary affect quaternary, right? So hydrogen bonds is in everything except primary. So hydrogen bonds are common all over the place, but they're also the weakest type of bond. And so just a little bit of heat, if there's enough of it, like enough to change to your body temperature from 98.6 to 104, 105, just a few degrees change, and that's enough energy that a lot of those hydrogen bonds aren't going to be so stable anymore. And we need hydrogen bonds all over the place. So heat can denature by breaking those bonds. Acid bases, they break hydrogen bonds between polar R groups or disrupt ionic bonds. Why? This is what I was talking about before. That's why I spent so much time talking about those ionic and polar amino acids at physiological pH 7.4. They are charged at 7.4, but you change the pH and what could you change? The charge. You could turn neutral into positive or positive into neutral, negative into neutral or neutral into negative, right? And without those ionic interactions, without those salt bridges, we're going to have the same shape? No. Agitation? Sometimes all it takes is just shaking them, stirring them, in a manner that's enough that it does pretty much the same thing as heating does. You're agitating enough to where those hydrogen bonds are going to break and stretch, changes the shape, and if it can't just naturally fold back into the normal shape, then...
So meringue and
broke.
scrambled eggs are considered agitated, or are considered denatured?
Mm-hmm.
Huh.
You're changing the cross linkages of the proteins. It's considered denatured in the same way that cooking an egg would be. Although, is meringue a sugar solution, or is there egg in there?
Oh,
There's
it's all egg.
right? Yeah, it's all egg.
It's all
Yeah.
albumin
Yeah, yeah.
and air. I thought it was like physically. They talk about it like it's causing them to latch.
Right, but the way it latches is by
It's chemical, it's not
stretching
physical.
it up. Yeah, soft stretching it off enough that They'll latch onto each other instead of onto themselves, right? So, application of denaturation. Here's what a normal egg looks like. Here's what a cooked egg looks like. Cooking it, adding heat, just causes it to those proteins to stretch out, and then they start cross-linking with each other all over the place, which is why instead of having a nice clear albumin here, you get that nice white albumin because those proteins are just cross-linked all over the place in there.
Don't add salt until the very end as well, because you'll end up with runny.
Huh.
Pulls water out of the.
Yeah, sure. Interesting. Cooking tip that I had never heard before. But it does make sense. What are we at? 41. Yeah, I'll wait. I have a thing, very topical here, to do with when I was in college and I was a member of a group that allowed drinking. So we had to go to an alcohol safety thing.
Teach you how to drink right?
No! They did something like I wanted to walk up and slap him at the end, but only because I was a biologist and chemist, so it's just like, that was bad. Anyway, we'll come back to it. Heat, acids, organic compounds can do it, like ethanol. Why does ethanol cause a problem? Because it interacts and interrupts the hydrophobic interactions. Hydrophobic interactions is one of the things that give protein shape as well. By forcing the hydrophobic cores to hide out away from the water, you throw in something that can associate with those hydrophobic cores, and it gives them something to dissolve in. So they won't be quite as tightly grouped anymore, and that could also cause a change in the shape of the protein. Heavy metal ions can interrupt disulfide bridges. They can interrupt some of those salt bridges by giving them something else, positive or negative, to associate with instead of that positive or negative charge on that other bit of the protein. Agitation by breaking the hydrogen and hydrophobic bonds, stretching it out. Meringue right there. All right, so that's how you denature proteins. Now let's talk about a special class of proteins called enzymes. Enzymes are catalysts. What is a catalyst? We talked about it before. Where did catalysts come up? They were mentioned in passing a couple of times, the most recent of which...
Acid catalyst.
Acid catalysts, yes, for the formation and breaking of ester and amide bonds, right? And acid catalysts were mentioned, although to a lesser degree, in how we did our biochemistry for the breaking of fatty acids as well, right? So, catalysts are things that take part in a reaction but are not inherently changed in the reaction. They are the thing that gives the structure for the reaction to happen. When we were talking about the hydrogenation of fatty acids to make more saturated fatty acids, we had a nickel catalyst, right? Nickel was the substrate that that double bond could come down to and interact with the hydrogen gas. It stabilized those two molecules together so that they could interact with each other and hydrogenate that bond. Well, in the catalyst here, when we're talking about enzymes, we're talking about something that's very similar. We're talking about a large structural molecule that will allow for a reaction to occur, but the important thing here is it cannot make the impossible possible. It can make the impossible, or excuse me, the possible more likely. It cannot make the impossible possible, but it can make the improbable probable. How's that? Your paper is not bursting into flame right now, because the activation energy of the reaction of not CO2 and water, but sugar and oxygen is high enough that the vast majority of molecules in your paper right now do not have the level of energy that they need to interact with oxygen in a manner that allows it to combust. But that exact same reaction is happening inside your cells at 98.6 degrees. Now, is that hot enough that your paper would start burning? Does your paper burst into flame on a 98-degree day? No. But it is hot enough that in your cells, in the presence of an enzymatic catalyst, those reactions can occur. And how does that happen? And this is how it happens, although we don't really get into exactly how it works. It just says it increases the rate of the reaction by lowering the energy of activation. To explain this concept, I'm going to go back to the potentially inappropriate metaphor of what it takes to shake someone's hand. Do we all remember this metaphor? All right, so now let's pretend that I take two of you and I blindfold you, because enzymes are blind, they do not have intention, right? And I tell you to stick your hand out in front of you because that's the shape that you occupy. What's it going to take for you to shake someone else's hand? The only other person in the room, also wearing a blindfold, also sticking their hand out in front of them, but me and one other person, we're going to spin you guys around in circles and then let you go and just tell you to walk around the room until you shake someone's hand.
Wait to float an aqueous solution?
Well, the room emulates the aqueous solution, yes. Eventually it might happen, but we have to wait for you to bounce around the room until you run into each other in just the right place so that when you grab the other person's hand, you can shake it, right? Now, what could we do to increase the likelihood of that reaction happening? Well, we could top you up on some sort of stimulant so that you run around the room a lot faster, which increases the rate at which you might come into contact with the other person, which increases the rate at which you might be able to hit them in the right orientation. Doesn't guarantee it, but increases the chance, right? Or, if one of us decided to take pity on you, what could we do?
Add more people to increase the size of the room.
I could add more people, yes, which would increase the number of collisions so that the number of correct collisions increases. Or I could be nice and I could take one person's hand and hold on to it until I grab another person's hand and hold on to it, and then when I have two hands in my hand, I could bring them together to make sure that they shake in just the right way. That's what an enzyme is doing. Now, in this metaphor, I would still be blind, reaching out for hands. But the difference is, when I find one of you, I will hold on to that hand until I find another person, and then I will bring those hands together in exactly the way it needs. So I would be the enzyme. And once they have shaken hands, I would let go, because my job is done. But I would still be there to find two more people and bring their hands together. The reason it lowers the activation energy is because normally those molecules have to come together in just the right way, with just enough energy. And then, and only then, will that reaction happen. But an enzyme allows one of them to come into the enzyme, and then the other one to come into the enzyme, and then the enzyme is going to bring them together in just the right way. So we don't need nearly as much energy, because we're forcing the reaction to happen just perfectly. They are molecular chaperones. Or to use more chemical terms, they create a microenvironment that lowers the activation energy required for a reaction to occur, hopefully below the amount that the molecules already have. You can still cool off your body enough to where the burning of sugars will no longer happen. Where even those enzymes won't be enough to make it happen. But at body temperature, those molecules already have more than enough if they come together in just the right way.
Don't some enzymes force a shape change in one protein to expose the active sites?
Yes, we'll get to that. We'll get to that. This is an example not so much of burning sugars, but of carbonic anhydrase. This enzyme catalyzes the combination of CO2 and H2O to form carbonic acid and a hydrogen ion. But remember what we know about equilibrium? In your body, we're going to really spike the CO2 concentration, right? Because your cells are making a lot of CO2. which is going to force the creation of a lot of this side, right? But in your lungs allow that CO2 gas to escape, which means that the CO2 concentration goes way down, which means this reaction is all of a sudden going to do what? Shift to the left again, breaking down a lot of this carbonic acid to reform CO2 that then escapes in the lungs. The exact same enzyme that absorbs that CO2 to make more of that buffer in your blood is the same one that catalyzes the reverse reaction to release the CO2 in your lungs. Who's paying for that energy increase that it takes to go in the opposite direction? Entropy. Our favorite friend behind the curtain. Because there is no such thing as a free lunch. Point is that your lungs aren't suddenly getting warmer in order to release that CO2. So somebody has to pay for that difference. But once again, it's not nearly as big of a paycheck as you might think, because the same enzyme that lowers that activation energy for this reaction also lowers it for this one. So how do we know that something is an enzyme? How do we name them? Well, the most obvious thing is anything with an ACE at the end. We're going to take the reaction or the reacting compound and put ACE at the end. So if we have something that, like sucrase, is an enzyme that catalyzes the reaction of sucrose. But it uses sucrose, but we don't know what type of reaction is being catalyzed there. So we have to add something on. It might describe what type of reaction is being catalyzed. So sucrose oxidase would be something that oxidizes sucrose. Or we might just have a common name that makes no freaking sense at all. Or maybe they do make a little bit of sense. Like, for example, pepsin. What do you think that does? Pepsin sounds a lot like peptide. What do you think pepsin does? Maybe breaks peptide bonds to help digest proteins. Trypsin does the same thing, but is not nearly as obvious. So we can classify them by what type of reaction they catalyze, like octoreductases, transferases, hydrolases, lysases, isomerases, ligases. These are all obvious things to me, but that's because I deal with these guys. So what do? An octoreductase, oxidation reduction, right? So do you think we're going to find a lot of octoreductases in, say, the Krebs cycle? It's all about redox reactions to make CO2 and burn sugars? Yeah. Transferases, transfer groups of atoms. These are things like kinases or phosphatases are aspects of transferases because they add or subtract phosphate groups. We have acyl transferases that add or subtract acyl groups to different things. Hydrolases. We've talked about a lot of hydrolases so far. The things that break amides, the things that break esters, the things that break fats. These are all hydrolases. They hydrolyze water to break apart ester bonds, to break apart amide bonds, right? Isomerases. When have we talked about isomers?
Way back when.
Way back when. In the chapter about...
The arrangement of the portions of the molecule.
Arrangement of portions of the molecule. Specifically, sugars, D and L isomers, right? An isomerase would be something that could rearrange a molecule, like take L-glucose and turn it into D-glucose. Take alpha-glucose and turn it into beta-glucose. And ligases are enzymes that combine small molecules together. DNA ligase would be an enzyme that takes multiple chunks of DNA and covalently glues them together. So an amino-acyl ligase would be something that fuses amino acids together, right? How does it do it? Well, one tiny part of enzymes is called the active site. And we call it the active site because that's where the action is happening. That is where the substrate will bind. And in this case, it is a substrate that's, let's say, sucrose. And it binds to the active site, and the enzymatic action of that active site hydrolyzes that sucrose into glucose and fructose, where it then releases it so that it is open and available for another sucrose molecule to attach. The active site is the location where the substrate binds. It's the location where the chemical catalysis happens. It's where the reaction happens. But I want you to appreciate something. The active site is the most important part of an enzyme with respect to the job that it needs to do. But that does not mean that the rest of the enzyme is worthless. If you change the active site, you can change whether or not the substrate will bind. Yes. But if you change the rest of the protein, that substrate still might bind. But now nothing happens. Because the enzyme has to do something in order to catalyze the reaction. And the rest of that protein is also necessary for any of that extra stuff to happen.
Do you think an enzyme can act as an inhibitor?
I'm saying that something that changes the shape of the enzyme can act as an inhibitor, yes. An enzyme itself, yes, can act as an inhibitor of another enzyme.
But I mean, if you had the form of the enzyme that didn't work,
Then it would
floating
not do the job.
around,
Yeah.
but it could bind the available things and render them unavailable.
For the real enzyme to do the job of them. Yes.
Okay.
Now eventually they would also float away, and then they might, next time they run into somebody, it might be the real one, or it might be another fake one. Those are called dominant negative versions of proteins, and that's actually something that's frequently done in experiments. You add a dominant negative version to slow down the reaction to determine whether or not that reaction is the thing that's actually doing the thing you're looking for. I just don't want you to think that the active site is the only important part of an enzyme. It is frequently the most important part, yes. It's frequently the thing that we target first, yes. But the rest of the enzyme is not just some sort of thing hanging off the side. It does something too. A hammer in my right hand is not going to pound very many nails if my right hand is the only thing that's there, right? Chop it off at the elbow and that hammer is pretty worthless. It takes the rest of me to actually pound the nail, right? So, why is the active site work? Why is it selective? Well, it's because when we have our substrate, the same things that cause the shape of the enzyme, right, can also cause the association of the enzyme with whatever the substrate is. A serine right here might associate with another serine, so there might be some hydrogen bonding. We might have a pocket of hydrophobic amino acids that therefore can shelter a hydrophobic amino acid on the substrate, or maybe a hydrophobic side chain on the thing if it's not another protein. We could have some salt bridges and a couple of guys that help to stabilize because we have a negative charge there and a positive charge there, so we can hold it in shape because of those associations. If we change any of those things, suddenly that substrate might not stick here, might not fit here. And now that protein can't do its job. We pretty much talked about all of this already. Region where the enzyme fits the substrate, contains amino acid R groups that help to hold it, and then when the reaction is done, it releases it. It is very rare for enzymatic catalysis of a reaction to involve covalent linkage with the enzyme. There's a very rare thing where the substrate is covalently linked to the enzyme during this reaction. And even if it is, to be an enzyme, that covalent linkage would have to be undone before the reaction is finished. Because the enzyme has to be complete in its original form at the end of the reaction, or it's not an enzyme, it's just another reactant. Make sense? So, what this leads to is when we're talking about enzymatic reactions, we can draw them out like this. The enzyme plus the substrate goes to form a reversible reaction between that and what's called the enzyme-substrate complex. And once that has formed, it will catalyze the release of the product. But it is also possible that it could simply re-release the substrate without doing anything to it. So this is reversible, and in this example, this is not. But we already have an example of something where it is, right? Remember when we were talking about carbonic anhydrase? The thing that takes CO2 and water to make carbonate and a hydrogen ion? In that case, there would also be another arrow like that. Because it could take the products and turn them into reactants to go in the opposite direction. As an example of breaking down a disaccharide in the case of lactose instead of sucrose, we bind to the active site. We are held together in specific bonds that position the glycosidic bond of that lactose in a very specific location that allows for R groups on the amino active site to catalyze the hydrolysis of that bond. And the products are no longer capable of being attached, so they're released. So we get lactose plus lactase, because it's the enzyme that works on lactose, right? Form the complex, and then that releases lactase in its original form, but now instead of lactose we have glucose and galactose. Leaving our enzyme free to do it again. And again, and again, and again, and again, and again. So there's two different models, two different metaphors about how this works that we could talk about. One of them is called lock and key. So it's like the key to a door, right? You have a door, it's the whole door is the thing that keeps you from getting in, right? Not just the handle, the whole door. If the handle's there by itself, you could still walk right in, right? But, so that's the whole shape of the protein. But the door is where the active site is. The handle, excuse me, is where the active site is. So we have this key, we can put the key in the active site, and if we have the right key, then it can associate with that active site and cause a change that opens the door. The metaphor isn't great because what we actually want is to change the key here, right? And doors don't change keys. But the lock and key model is a good metaphor for having just the right key to fit into just the right shape of the lock. And then once it's in there, there is a change. It's a very static model, though. And so the lock and key model is a nice metaphor for how proteins interact with their substrates, how they are specific for their substrates. But there is a better one. And that's the induced fit model. In this example, the enzyme isn't this structurally tight, perfectly always shaped door handle. It has an active site that's more flexible because enzymes are flexible. Protein chains are flexible, right? But as we bind the substrates, it brings together that active site. How could it do that? Yeah, sure. Let's go back to this. So, here we have a positively charged NH3 group right here, right? Here we have a serine with a hydroxyl group. We know that that hydroxyl group can form hydrogen bonds, right? So it's partially negative. That oxygen right there is partially negative. So if we wanted to, that negative charge right there could sort of maybe associate with this positive charge down here, right? But it's a little bit too far away, maybe. It can't quite do that. But if we bring in a substrate, and then of course we have this this hydrophobic bit over here, right? That has no reason to associate with these two guys at all, so it's just hanging out there. But if we bring in this sugar, it will lock into place because of that hydrogen bond there and because of that salt bridge there. And now all of a sudden we have a hydrophobic thing right here that's kind of close to all those hydrophobic things there. And what do hydrophobic things like to do? Entropy is going to pay for them to hang out together, right? And so suddenly that thing that was hanging out there that had nothing to do, suddenly it has a group that it wants to hang out with. And so as the substrate binds in the active site, that can induce the fit, induce the wrap around of the enzyme so it comes around together in just the right way. And if the thing it's bringing together is actually a guy right here that's hanging out into this space that's going to go in here and cut any bond that it finds, then that's how we get our hydrolysis of sugar. If this was glucose and this was galactose, then by bringing in our sugar, locking it into position, drawing in this other side, it brings together the things that actually do the cleaving. And once it's cleaved, these two things don't sit here anymore. This can float out, that can float away because it's not locked together, and so it doesn't hold its shape anymore. So it has to open back up again and let those guys go, leaving it open and ready for the next molecule that comes in. That's the induced fit model. The enzyme is flexible. It adjusts the shape of the active site as the substrate binds. That means that it can have more than one key. More than one thing could fit into that thing, but it may not actually mean work is going to happen, but more than one thing could possibly fit there. As long as it holds on to those important things and attracts the other side, it's not going to be just one key, but it's still going to be a very limited set of keys, right? And more importantly, it implies that the shape of the entire protein could conceivably change during this reaction. The active site is the place where the substrate binds, but the entire protein might have to change shape in order to do the job. This is attempting to illustrate the induced fit model without actually changing the shape ever in the drawing. So I don't know how good of an illustration that actually is, but here we have glucose and galactose binding to the enzyme's active site. It has to bind in a very specific orientation. Why? Because enzymes are made of chiral amino acids, which means that they will only bind sugars that have a chiral shape, right? So they can only bind things in a very specific orientation, but if it does find something with that shape, it can catalyze the hydrolysis of that diester bond, or the ester bond, excuse me, glycosidic bond, and produce galactose and glucose. So, does anyone have any questions about why the shape of an enzyme is important? Denaturing enzymes. Obviously, all of this is very concept-based, right? Done a lot of math so far in this chapter?
Don't say so far, because it implies that there might be some.
So far. All right, let's talk about isoenzymes. Isoenzymes refer to isomers of enzymes, right? So isomers, before we're talking about D-galactose, L-galactose, alpha, beta, right? Different isomers that have slightly different shapes. Well, there are enzymes that can have slightly different shapes but still do the job as well. Different forms of an enzyme that can otherwise catalyze the same reaction. They come from different quaternary structures. So that means they're using different proteins, different subgroups, so different tertiary structures, right? That will form a different multi-protein subunit, i.e. quaternary structure. that still does the same job. So by example, there's this thing called LDH. So when you were working as a nurse, did you do LDH assays? Did they have you do those? Or did it ever like tell someone the LDH results?
No, most of the time you don't tell anybody results. They want the doctor doing that most of the time.
Gotcha. Well, I mean, did
just
you
to
ever...
take us out of.
Gotcha. Did you ever tell the doctor though?
Yeah.
Right. So here's how this works. Lactate dehydrogenase. What's that do? It does something to lactate, right?
Turns it into pyruvate.
Yeah, how does it do that? It removes a hydrogen. And it's an enzyme. How do we know that? Because it's got ACE at the end, right? So it's an enzyme that removes hydrogen from lactate, resulting in pyruvate. It takes away this hydrogen right here and that hydrogen, causing a double bond between that carbon right there. So lactate dehydrogenase is an enzyme that takes lactate and turns it into pyruvate, which is usually then shuttled into their Krebs cycle.
And we get a free NADH out of it.
And we do, we get NADH out of it. So, there are 5 isoenzymes of LDH. They can all do the same job. But each LDH isoenzyme uses different subunits. So there's the M and the H. The M subunit and the H subunit. In the liver and the muscle, you get LDH that is all M4, so we call it LDH5. In the heart, you get the same reaction for the same purpose, but there you get LDH with H4 isoenzymes, so all 4 H subunits. Same job, just as good, but we're using different subunits to do that same job. Why am I talking about it? Well, here's the thing. When you have something like a heart attack, where you have damage to a lot of cells, when those cells die, they will lyse and release a lot of their contents into the bloodstream. Another thing that works on this, before we actually get into the description there, is creatine kinase. It's another isomerized enzyme. If we have a heart attack, then that means we're going to release a lot of H4 LDH. If we have liver damage, then we're going to get a lot of M4 LDH. So you can look at blood levels for these enzymes and it'll tell you where is the damage. What's going wrong? A simple blood test can tell you if there's something wrong with the heart where we have cells dying and lysing, where the liver is having problems and is lysing and a lot of the liver enzymes are being released into the bloodstream. So here we have an example of some of the different things. H4 is heart and kidneys. 3H1M is red blood cells. A little bit from heart, kidney, or brain. We get H2M2, brain, lung, and white blood cells. H1M3, lung or skeletal muscle, and M4 is skeletal muscle and liver. So an LDH test, LDH blood test, can tell you if there's some systemic problems and give you an idea of where those systemic problems are occurring. So the point of that little section was just to let you know that there's more than one way to skin a cat, and that how you skin a cat can tell you what's going wrong, right? Different isomers of proteins. If those proteins are isolated to specific regions of the body, they can be biomarkers for problems. So what can affect how well enzymes work? We have this picture here of a lovely hot spring. Temperatures are 50 to 120 degrees Celsius. What would happen to your hand if you put it into 120 degrees Celsius? What, could you put your hand in 120 degrees Celsius water?
Yeah, nobody stop you.
You burn yourself. But
120 degrees
Burn it
Celsius
off.
water would be called steam
You wouldn't quite
at
get
one
that.
atmosphere. But yeah, if you put your hand in, let's say 100 degrees Celsius water, what do we call the process of putting live things in 100 degrees Celsius water?
Stupid.
Boy.
Boiling.
Boiling. We call it cooking them,
Okay.
right? Which would imply that they can't survive that process. Except that there are bacteria that can. There are bacteria that not just survive, but thrive at those temperatures. And what would happen if you pulled them out and tried to grow them at room temperature? A whole lot of nothing. They would die. Most of them would die. Some of them would just go inert until they got nice and warm and toasty again. Same thing that happens to us. When we get too cold, too far below our optimal temperature range, we die. These guys have evolved to live in environments that have really, really high temperatures. So the point is that 100 degrees Celsius does not automatically make all proteins suck and not function. It just means that you have to make those proteins at those temperatures so that they hold the shape that you want at that temperature. So, it implies that any enzyme is going to have an optimal temperature to function. This implies that it's always going to be 37.5. In humans, it is. Actually, even that's not true. Have you guys heard of something called heat shock proteins? It's one of the things that you can test for whether or not there's been a lot of damage, whether or not something has been overheated recently. Heat shock proteins, you have genes that you don't normally express, but as the temperature starts to rise, you will start turning on these genes. Because they are versions of genes that you need that will either do a job that a normal protein can't do because it's too hot, or they will help the normal protein hold its normal shape when it should just melt and denature. But even then you get too high, and even those will fail, will start failing, and then you will die. There are other organisms that don't have as many of these, and so they would be much more susceptible to changes in temperature. pH. Just like there's an optimal temperature for a given enzyme, there is also an optimal pH for a given enzyme. Now for most of the ones in your body, that optimal temperature is probably going to be the normal physiological pH, right? 7.4. Why is that so low? 7.4. It should actually be just a little bit further up from that 7.4. Optimal pH for most, just above 7. This is a logarithmic scale, right?
Is that on the wrong side of the line?
No, 7.4. Higher than 7.
Okay.
If we change that pH, why does that change the effectiveness of enzymes? Because it could change the positive negative charges of some of those side groups, which would change their positive negative association, right? Which could change their shape. And shape is what matters. Now, there are areas of your body that do not have a pH of 7.4, right? Pepsin is an enzyme that works in your stomach. It's made by the cells lining your stomach. Why does it not just digest all of those cells as they make it? Because it's an enzyme that doesn't work at a pH of 7.4 inside the cells lining your stomach. But when they export it into your stomach, where the pH drops all the way to 1.5 to 2.0, all of a sudden they take a shape that is much more functional. And they will digest any peptide bond that they run into. Lactase, sucrase, they work in the small intestine. They have a different pH here. Amylase from the pancreas. The liver works at urase from the liver, has a pH about 7, 7.0. trypsin in the small intestine works on peptide bonds. It actually likes 7.7 to 8. But that lower area of the small intestine becomes rather basic as opposed to the very acidic stomach, if you recall. Arginase in the liver works at a pH of 9.7, so it actually likes it to be pretty basic. And then we get to the idea of inhibitors. So before, we talked about things that denature the enzyme, right? So we have optimal temperature because too low, too high can denature the enzyme. Too low doesn't denature it so much as it just can't do its job because the substrates don't have enough energy to react, even in the enzyme. Too high leads to denaturation. pH, too high, too low, can denature. But now we're talking about inhibition. So inhibition is a concept in a biological reaction that happens even at ideal temperature, ideal pH. Inhibitors prevent the enzyme from creating the product. Anything that prevents the enzyme from doing its job under otherwise ideal conditions is considered an inhibitor. But there are two different types. There are competitive inhibitors and non-competitive inhibitors. Competitive inhibitors are stand-ins for the substrate itself. They're something that will bind to the active site just like the substrate does. The difference is they aren't the substrate. And so it's like handing a paper to a kid with those craft shears so that they can't cut their fingers off. Except you didn't hand a kid a paper, you handed him a very thin sheet of polyurethane. He's just going to sit there for a while and get pretty pissed off. Because those shears are not going to cut that. And as long as he's trying to cut the fake stuff, he's not going to be ruining your homework. It's a new phrase for non-traditional students. The dog didn't eat my homework. My son drew pictures all over it. Or my daughter used it in her craft project.
So homework should be done on polyurethane sheets.
Or online.
They're dog-proof.
So, substrate plus enzyme-substrate complex goes to make the product. But if you have an inhibitor, the inhibitor is going to bind to that enzyme, and for every inhibitor that is bound to the enzyme, it cannot bind to substrate. Now, that substrate can still fall off without being catalyzed. The inhibitor can still fall off. But if it falls off, it might run into another enzyme and then block that one instead. But the point is that if we have a whole bunch of inhibitor around, if our inhibitor levels are higher than our substrate levels, we could reach a point where we will completely saturate that enzyme with inhibitors, virtually stopping that reaction. Where would you want to do that? And the answer is
Everywhere.
a significant majority of all of the drugs that someone might take for fill-in-the-blank condition. Many of the drugs that we give people for different treatments, the reason those drugs work is because they inhibit a reaction. They stop something from happening that for whatever reason we don't want it to happen as much. Antihistamines. They're competitive inhibitors, most of them, are competitive inhibitors for the histamine receptor. They will bind to the histamine receptor, but in a manner that does not actually trigger it. But it does block it from being activated by normal histamine. And so those plants can be having an orgy in your nose, but your nose won't realize it. That's what's happening with pollen. It's sperm.
Oh, jeez.
Pollen in your nose, the reason we react to it is because of the invasion of sperm from the pollen grains.
I got a 15-second comprehension delay right now.
Yeah. Once a year, a friend of mine makes a Facebook post. I wish all of those trees would get a room instead of having an orgy in my nose, because he's allergic to aspen pollen or something like that. Just really sits up his hay fever. Which releases histamine, which the rest of your body responds to, which is why you sneeze and have a watery nose. Antihistamines block that reaction. Pollen's still there. Your cells are still going, what the? But the rest of your cells are going, I have this neat little guy. He knocked on my door. He hasn't left yet, but I'm talking to him. Who are you? Instead of realizing that histamine's floating around, screaming and running around with their head cut off, going, we're being invaded! Most antihistamines are competitive inhibitors.
But then what if some people are just unaffected by it?
Resistance is a thing. Here's a graphical example of competitive inhibitors, or more visual as opposed to graphical, I suppose. Active site plus substrate. Instead, if we add an inhibitor that also fits that active site, it will prevent the substrate from binding, which means that we get no reaction, we don't get a product. And if that product is a reaction of a cell to the presence of histamine, then we don't have hay fever. If that product is a chemical component that is causing a problem, then we don't have that problem. A lot of the drugs that we prescribe are inhibitors. Many of them competitive inhibitors for specific reactions that we want to prevent, to stop an otherwise inappropriate reaction or condition from occurring. All right, so if there is a competitive inhibitor, what about a non-competitive inhibitor? The whole point of a what does a competitive inhibitor compete for? Why do we call it competitive?
Because there's a finite number and
a finite number of what?
Of
a very specific part of a protein that does work.
the active site.
There's a finite number of active sites. We are competing for active sites with the substrate. So a non-competitive inhibitor is still going to inhibit, but it's not going to be competing for the active site. A non-competitive inhibitor attaches somewhere other than the active site, binds to an enzyme at a site other than the active site. In so doing, it distorts the shape of the protein, which means we also change the shape potentially of the active site. Even if we don't change the active site, we do change the shape of the rest of the protein. So even if something does stick, protein can do its job. So we still don't get the product. So it still prevents the creation of the product of that reaction, but it does so by sticking to the enzyme. It doesn't block the active site. It just blocks the protein from doing its job. That's non-competitive. So the difference is, for a competitive inhibitor to block a reaction, we have to give enough of it that we have way more of it than the substrate. We have to make sure that the most likely thing sticking is the inhibitor, right? Because in a competitive inhibitor, if the normal substrate sticks, it's going to react, right? But in a non-competitive inhibitor, we don't need to give a bunch of it. We just need to give enough of it that most of the enzymes are going to react to it. Now it doesn't matter how much substrate is there. As long as our non-competitive inhibitor is there, it's going to prevent them from doing anything. Even if we add more and more and more substrate, it doesn't matter, because as long as we have enough non-competitive inhibitor to shut down all the enzymes, nothing's going to happen. But if we added more and more and more substrate to the competitive inhibitor, eventually the substrate would outcompete, right? And then we would still see the products. And then we get irreversible inhibition. We've talked about one other example of irreversible inhibition before.
Carbon monoxide.
Carbon monoxide. Carbon monoxide is an irreversible inhibitor of hemoglobin because it covalently binds to the iron in hemoglobin and never lets go. Those competitive inhibitors, the non-competitive inhibitors, they bind, but then they fall off. They rebind and then fall off, and then rebind and then fall off. Not to the same one, to a different one, same one, somewhere else, doesn't matter. But the point is it's not permanent. That's why even with the non-competitive inhibitor, we would still want to give you more than a 1-for-1. We don't give you one molecule of inhibitor for every one receptor that you have. We want to give you a lot of it, but we don't need to give you nearly as much as we would have for a competitive inhibitor. Well, these guys, they just shut you down. They irreversibly block a reaction. And so the only way that you can recover from that is to make new enzymes. The only way you recover from carbon monoxide toxicity is to make new red blood cells, or get them from somebody else. Often a toxic substance because it destroys the enzymes. Usually does so via covalent modification that prevents catalytic activity. An example of this, nerve gas, insecticides, or antibiotics. Penicillin, penicillin G, V, ampicillin, amoxicillin, all of these guys, they kill bacteria because they irreversibly block the enzymes that build the bacteria's cell wall. And without that cell wall, those cells can't grow, they can't multiply, and they are really vulnerable to immunological attack. So how would one develop penicillin resistance, which is very common? What if you made an enzyme that breaks penicillin? Penicillin resistance comes in the form of bacteria that make an enzyme that will break pretty much all of these, and they export that enzyme into their environment. So they make a guy that breaks the antibiotic, and then they put it in their environment so that it gets broken before it even gets to the cell. So it can protect the enzymes that make the cell wall so they can do their job so the bacteria can survive. Why does penicillin do nothing to you? Do you have any enzymes in you that make cell walls? Nope. So, would it do anything in you? Nope. Why are the nasty antibiotics that we're having to develop now so nasty? It's because a lot of the processes that are being blocked in bacteria, you also sort of do those things. And so it has side effects in us too. Competitive, non-competitive, or irreversible. Irreversible does not have similar shape to the substrate, forms covalent bonds that modify the enzyme, and it is permanent. And that's the chapter. So we're going to pick up with the next chapter and ostensibly last chapter Tuesday. And we'll take our time walking through it. It's got like 80 some odd slides. I just posted it tonight. So, you know, we'll take our time, but we'll probably get most of the way through it, if not all the way through it. And then Thursday, we will finish it. And then we'll have whatever time you guys want for discussion, more in-depth material, questions about anything else, up to and including questions from chapter 5 or chapter 18, if you have them, and we could just kind of chat about them, but it won't be something I'm going to put on the test. Both of them are interesting chapters. I will understand if your life does not allow you to fully entertain the possibility of learning from them. So, I will hopefully have a homework posted for you guys that will hopefully go live tomorrow morning. Got some extra sleep tonight, so should be able to get that done pretty quick. Or I got some extra sleep this morning, I should say. So I will see you on Tuesday.
I just started
Wait, what?
next week. Next Monday.
What?
Tuesday, Wednesday? Or
Tuesday.
Tuesday.
I
Yeah,
will see you
Tuesday.
on Tuesday.
I'm losing it.
Class on Tuesday. Right? Class?
Yes.
On
Yes.
Tuesday?
Oh, today's Thursday.
Yeah.
Today is Thursday.
I keep thinking today's Wednesday. I have too many classes, it's too much fun.
So we're gonna finish you Thursday.
Just just downloaded some information to statistics that I can. My brain goes faster than my fingers.
You're falling into one of the traps I fall into, which is conversational English. You can hear the attitude. Student is
try
anxious
not-
enough in asking for help to begin with,
That's why
much
you're
less having to.
That's why you're editing it.
Yeah.
I'm trying not to, just make it professional, not
Right.
attitude. Because I meet with her in the morning. I've already met with them, but like I just got an email from him because he still didn't do it, and it's like, are you flipping kidding me?
Well, you shouldn't communicate with him anymore.
Well, I'm not. I just sent him an email going, look, it's still not. He did one but didn't do the rest, and I'm like, because he told me, well, why do you need it? You haven't used it before.
That's
And then
none
he-
of his damn business.
I know.
Okay.
And then he said, well, what's her name has it too.
Oh my gosh, you broke confidentiality.
Yes.
And
I was crying.
And you said
crying.
you singled people out? Okay, Mr. Professor,
Yes.
I'm gonna turn the recording off.